Hello, Otters here, and welcome to another Ossett History podcast. And this time we're looking at significant figures and events from medicine from the Renaissance to the present day. And at least one, probably two, if not more, of the people and the related events, discoveries that I'm about to talk about will come up on your exam. So it's very important that you have a good, in-depth knowledge of what happened. We're going to look at Vesalius, William Harvey and the printing press first. And both of the men, Vesalius and Harvey, lived during the Renaissance, which, roughly speaking, lasted from 1500 to 1750. Vesalius was the first person to really question Galen's ideas, particularly when publishing his book, The Fabric of the Human Body, in 1543. He used dissections to prove Galen wrong, and one example of his claims was that he found the human jawbone to be just one bone, rather than two that joined in the middle, as Galen had claimed. Galen had been wrong because he had based his ideas on the dissections of monkeys and pigs, whereas Vesalius used human bodies. William Harvey, just like Vesalius, published his own book, this time called An Anatomical Account of the Motion of the Heart and Blood in Animals, in 1628, which also suggested that Galen had been wrong. In his book, Harvey wrote that blood is not constantly used up by the body and replaced by the liver, as Galen had written, but instead he suggested that, correctly, that blood is repeatedly circulated around the body. Both Harvey and Vesalius' ideas were able to spread fairly rapidly, unlike the people who came before them, due to the crucial invention of the printing press. Despite itself not being anything medical, the printing press was one of the most crucial inventions in the development of medicine, due to the fact that it made it easier for people to reproduce books and other publications, and thus ideas spread faster. Now, going back to the things that Vesalius and Harvey had said Galen was wrong about, there were other examples of things that they said Galen was wrong about. You would have to look in your revision notes and in the textbook to find out what they were. Next, we look at Edward Jenner and vaccinations. Jenner made the first major breakthrough in medicine of the Industrial Revolution, when he found that not one person had died of smallpox in a village called Gloucestershire. And this was very unusual at the time, that not one person had died from it. He found that many people in the village caught a disease that was not deadly, called cowpox, and that this somehow made them immune to smallpox. He tested this theory by giving 23 people cowpox, and when subsequently none of them contracted smallpox, despite being exposed to it, he concluded that giving people cowpox would protect them from smallpox. He called the process vaccination after the Latin word for cow, vacca, or vaxa. I'm not sure. I'm no linguist. Jenner's work spread quickly, again due to the availability of the printing press at this time, but also because governments, or the British government in particular, was beginning to take their first tiny steps away from the policy of laissez-faire, which was the whole, it's not our problem, we shouldn't get involved sort of attitude. And the British government gave him £30,000 in total to help him with further research. The effects of Jenner's research were mixed. On the one hand, tens of thousands of people were vaccinated against smallpox during Jenner's lifetime, but the vast majority of people were not for a very long time. However, after 1872, when the British government enforced compulsory vaccinations, smallpox was practically wiped out in Britain. On the other hand, some doctors initially were reluctant to accept the new ideas. The Royal Society refused to publish them. And, perhaps most importantly, though Jenner could prove his ideas worked, he had no idea why they worked, and therefore the findings could only be applied to smallpox. Until our next important figure came along with a revolutionary discovery and another vaccination to boot. That other person was Louis Pasteur and his germ theory. Whilst working for the vinegar and brewing industries and using powerful microscopes, just invented, Louis Pasteur discovered that he could stop liquid going sour by heating it. He decided that this was because the heat killed bacteria, or germs, and in 1861 he published his germ theory. The theory of spontaneous generation was very popular at the time, and suggested that rotting flesh or vegetation produced harmful bacteria, harmful germs. Pasteur disproved this theory by putting meat in a sealed container. Normally you'd expect meat to degenerate, to start to rot. But he showed in the sealed container the meat did not start to decay. He concluded that therefore germs were in the air all around us and not produced by rotting vegetarian or meat. Pasteur also found a new vaccination when experimenting with chicken cholera. He accidentally left some chicken cholera on the side, exposed to air, and when he injected it into a chicken, the chicken didn't die. The chicken was then immune to chicken cholera. Pasteur called this process vaccination, in tribute to Jenner's work. To begin with, many people didn't accept Pasteur's work, but as more and more medical people accepted the germ theory, there were significant developments. And it was these consequences of the germ theory which was the biggest legacy coming from Louis Pasteur. One example of the impact of the germ theory was Robert Koch, a German scientist who began work to identify specific germs, and in 1875 he successfully identified the germs that caused anthrax in sheep, before a more important discovery when he identified the germs that caused tuberculosis, or TB, in 1882, and then the germs causing cholera in 1883. Koch also found that it was possible to stain specific microbes using chemical dyes, and therefore make it easier to identify and study them next to the thousands of other microbes in the air. The impact of Pasteur's work also had an impact on surgery as an interesting side note, because he influenced Joseph Lister in his development of carbolic acid. Carbolic acid was sprayed during an operation to kill germs and keep a wound antiseptic. Into the 20th century, and the combined work of Pasteur and Koch led to the development of magic bullets. Instead of identifying the germs that cause disease, Emil von Behring started work to identify the antitoxins in the body that fought specific diseases. He worked with a research team, a key development of 20th century medicine. The idea was to find a specific antitoxin and to combine it with the chemical dyes that attached them. Well, the dye attached itself to specific germs, as discovered by Koch. Then to inject the result into the body, the dye would attach to the antitoxin to the germ and kill it. A Japanese man came onto the team in 1909 called Dr. Sahachiro Hata, and he found that a previous test dismissed as a failure was in fact correct. It had been the 606th test, and the magic bullet which attacked and cured syphilis was called Salvarsan 606. Dr. Gerhard Domack, probably not pronounced right, but never mind, took this research further and discovered the second magic bullet, this time for blood poisoning in 1932. By the 20th century, the technology that was available for these new magic bullet pills to be mass-produced, and the pharmaceutical industry began to expand rapidly. Everything that we've just talked about there would have been completely impossible without the development of the germ theory originally by Louis Pasteur, so it had a massive impact on medicine. Now we're going to look at Fleming chain Florey and penicillin. And we're into the modern era now, the 20th century medicine. In 1928, Alexander Fleming noticed an unknown mould growing on a petri dish was attacking and killing some bacteria. He decided to investigate further, and found that the mould attacked and killed other bacteria as well. He published his findings in 1929, calling the mould penicillin, but his research was limited because he didn't test penicillin on living things. He just experimented in his lab, assuming with petri dishes and other types of bacteria, and see what the mould did. However, 10 years later, in 1939, two scientists called Florey and Chain read Fleming's research and decided to test penicillin on living things, first on mice, then in 1941 on people. A man was given penicillin when he was dying from infection, and it appeared as a miraculous cure. He was getting better. Unfortunately, the penicillin was difficult to produce, and it ran out. So the patient got worse again and died. Fleming discovered penicillin. Florey and Chain proved that it worked spectacularly on living things. But there was one huge problem. It was impossible at the time to mass produce penicillin. And think about it, it's mould. I mean, how could you go and produce mould? It happens, it grows itself. So it was a very difficult problem. This remained the case because at the time Britain was at war, it was the Second World War, and British factories were being bombed, and the government was committed to producing other drugs vital to the war effort, rather than investigating new drugs that may or may not work. Fortunately for the production of penicillin, unfortunately for the people living in Hawaii, Japan attacked America at Pearl Harbor and brought the USA into the war, and the US government was willing to pay Florey to research ways of mass-producing penicillin in America, correctly realizing that it could save thousands of soldiers' lives. So Florey went to America, experimented, and finally discovered that freeze-drying the mold successfully meant that penicillin could finally be produced on a mass scale. Penicillin was first mass-produced in 1944, one of the biggest medical breakthroughs of all time. And now to our final significant event on people from Medicine Through Time. And we're going to look at Crick, Watson, Wilkins and Franklin and DNA. When it became clear through a technique called crystallography that each cell in the human body contains DNA, which is kind of like a set of codes that determines things like eye colour, height and other such characteristics, it was soon seen as important to try to investigate DNA further, to try to discover the structure of it. Watson and Crick knew that if the structure was found, then it might be possible to identify the genes that were responsible for causing inherited conditions, such as Down syndrome. Crick and Watson used the work on X-ray crystallography of Wilkins and Franklin to investigate DNA. DNA is incredibly small, so they needed these special techniques like X-ray crystallography, which is a process that allows detailed images of incredibly small areas of the body to be created. Or in incredibly small areas, anything I suppose. Crick and Watson discovered the structure of DNA in 1953, and later, in 1990, Watson led the Human Genome Project to try to map the location of every single one of the 30 to 35,000 genes in the 23 chromosomes in every cell of the human body. A bit of a task there, a bit of a challenge. The first draft of the work was published in the year 2000. There is now a better understanding of conditions such as Down syndrome and leukemia, and whether people are more likely to develop certain forms of cancer. There are now techniques for skin grafts, better vaccines, and better insulin for diabetics is now available. Stem cells have also now been discovered. Stem cells are cells that can transform into other types of cell. There is a possibility that we can somehow use them to replace faulty cells around the body to help people with specific conditions or illnesses. Many of the consequences of the discovery of the structure and of the mapping of DNA have yet to be found. But if you go to the VTLE textbook page 51, you will find a diagram of possible future developments, from the possibility that we might be able to mould bone marrow cells to repair tissue damaged by heart attacks, to the possibility that we might be able to repair faulty genes that are responsible for breast cancer. And so ends our podcast on significant individuals and discoveries in medicine. We've looked at Vesalius and Harvey from the Renaissance, before looking at Jenner and vaccinations along with Pasteur and the germ theory from the Industrial Revolution. Finally, we've studied the significance of Fleming chain Florey and penicillin, and the discovery of the structure of DNA led by Watson and Crick. You now have a full set of Medicine Through Time podcasts to help you to revise for your exams, that together last roughly the length of a 1-hour history lesson. I fully recommend that you listen to these often, making notes as you go, but do not forget that you must also use your own revision notes, the VTLE textbook and any revision guides that you have bought if you want to reach your full potential. I've been Otters with another GCSE Medicine Through Time history podcast. Good luck with your revision.